Hello dear friends, welcome back to my channel. I am Professor Namrata Burate. We are going to continue the next part of the fifth topic, that's the eukaryotic cell. So, what's the characteristic features of the eukaryotic cell? They have well organized membrane bound nucleus and organelles. Presence of membranes give clear compartmentalization of a cytoplasm. They have complex locomotory and cytoskeletal structure. Their genetic material it is organized into a specific structure that says as the chromosome. In case of eukaryotic cell, there is a presence of the two specific form of the cell. First one is a plant cell and the second one is an animal cell. In case of the plant cell, there is a presence of cell membrane, cell wall, endomembrane system. It includes the endoplasmic reticulum, Golgi apparatus, lysosome, vacuole. In addition to these, there is a presence of mitochondria, plastid, ribosome, cytoskeleton, cilia and flagella. There is a presence of nucleus and the microbodies. In case of animal cell, there is a presence of the cell membrane, endomembrane system. It includes the endoplasmic reticulum, Golgi apparatus, lysosome, vacuole. There is a presence of mitochondria, ribosome, cytoskeleton, cilia and the flagella, centrosomes and the centriole, nucleus and the microbodies. The first cell organelle we see that says as the cell membrane. The chemical study on human RBCs shows that the cell membrane it is composed of the trilaminar structure, means it consists of the lipid bilayer and a protein layer. In addition to these, there is a presence of the carbohydrate. The lipid, it is mostly the phosphoglycerides, have outer polar head. So you can see there is a presence of outer polar head. This outer polar head, it is a hydrophilic in nature, and the inner phytol tail, it's a hydrophobic in nature. So the non-polar tail of the saturated hydrocarbon, it is protected from the aqueous membrane. In the cell membrane, the ratio of protein and the lipid varies in different cells. For example, in case of human RBCs, membrane has a 52% of protein, 40% of lipid. Based on the ease of extraction, the membrane protein are of the two types. First one is a integral protein and the second one is a peripheral protein. The integral protein, it is partially or the totally buried in the membrane. The peripheral protein, it is lie on the surface of the membrane. The next one is the fluid mosaic model, which is put forth by the Singer and the Nicholson in 1972. According to this, the cell membrane, it is a QC fluid nature of lipid enable the lateral movement of protein within the overall bilayer. This ability to move within the membrane, it is measured as its fluidity. The cell membrane, it is perform the function. The first function, it is a transport of molecule. The membrane, it is selectively permeable to some molecule present on either side of it. Due to the fluid nature, the plasma membrane can help in cell growth, formation of intracellular junction, secretion, endocytosis and the cell division. The next, the cell membrane, it performs a two specific type of function. The first one, it says as the cell membrane, it's of the two type of the transport. First one is a active transport and the second one is a passive transport. What's a active transport? The first one we see the passive transport. This passive transport, it is a mode of movement of molecule or the substances across the membrane. So there is the movement of the molecules or the substances across the membrane without the requirement of energy. It can be further divided it into the three types. First one is the osmosis, second one it's a simple diffusion and the third one it's a facilitated diffusion. So what's a osmosis? The This osmosis is the process by which the water molecule it is passed through a membrane from a region of higher concentration to the region of the lower concentration. The second one is a simple diffusion. In the simple diffusion, in this process, the neutral molecules move across the membrane along the concentration gradient. That is, the movement is from the higher to the lower concentration. For example, gases and the small molecules. The third one is a facilitated diffusion. In this process, the molecules are transported along the concentration gradient by the help of the ion channel and the permease. The energy is not required in this process. The second way of transport, that is known as the active transport. This active transport, it is a movement of molecule across the membrane against the concentration gradient. That is, the movement is from the low to the higher concentration. But what's a significant phenomena? There is a utilization of the ATP molecule. It occurs in the few ions and the molecules. Its example is the sodium and the potassium pump. The polar molecule, it requires a carrier protein of membrane to facilitate their transport across the membrane, because they cannot pass through a non-polar lipid bilayer. Afterward, there is the cell wall. The cell wall, it is a non-living rigid structure found outer to the plasma membrane of the fungi and the plants. The cell wall of algae, it is made up of the cellulose, galactans, amines and the minerals like the CaCO3. In case of other plant, it is consist of the cellulose, hemicellulose, pectin and the protein. The cell wall of the young plant, it is shows the presence of the primary wall. Then there is a presence of secondary wall and there is a presence of middle lamella. So what's this middle lamella? This middle lamella which is present, it is the thin lies between the two adjacent cells. It is the first structure formed from the cell plate during the cytokinesis. It is mainly made up of pectin, calcium, magnesium pectate. That is the softening of the ripe fruit. It is due to the solubilization of the pectin. The second layer, it says as a primary cell wall. This primary wall in young plant, it is capable of growth. It is laid inside to the middle lamella. It is only wall seen in meristematic tissue, mesophyll tissue as well as in the pith. The next one is there is a presence of the secondary wall. This secondary wall, it is present inner side to the primary wall. Once the growth of the primary wall is stopped, the secondary wall is laid. At some places the thickening is absent, which leads to the formation of the pith. That is the plasmodesmata. It is the cytoplasmic breach between the neighboring cell. It shows the pores between the cell wall and the middle lamella. Next, about the functions of the cell wall. The first function, it gives the shape to the cell. Second, it protects the cell from the mechanical damage and the infection. The third one, it helps in cell-to-cell interaction. It acts as a barrier to undesirable macromolecules. The next one is the endomembrane system. This endomembrane system, it is a group of the membranous organelles having the coordinated functions. It includes the endoplasmic reticulum. Secondaries includes the Golgi bodies. Next series include the lysosomes and the vacuoles. As the first phenomena we see, it's a endoplasmic reticulum. This endoplasmic reticulum, it is a network of tiny tubular structure which is scattered in the cytoplasm. So this one is the cytoplasm and this endoplasmic reticulum, it is scattered in the cytoplasm. Endoplasmic reticulum divide intracellular space into the two compartments. The first one is the luminal site, that is inside the endoplasmic reticulum compartment, and the second one is the extraluminal compartment that is present in the cytoplasm. This endoplasmic reticulum, depending upon the nature of its membrane, it is of the two types. First one is the rough endoplasmic reticulum and the second one is a smooth endoplasmic reticulum. So this one is a smooth endoplasmic reticulum. The first one we see, that is the rough endoplasmic reticulum. This rough endoplasmic reticulum, they are found extensive and continuous with the outer membrane of nucleus. They have rough membrane because they bear the ribosomes being attached to their surface. They are actively being seen in the cell, which have their involvement in the synthesis and the secretion of the protein. The second one is the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, these are smooth because they do not bear the ribosomes in the form of granules on their surface. It is present in the cell where they act as a major site for the synthesis of the lipid, also help in the synthesis of the steroid hormone in the animal cell. As is the endoplasmic reticulum, this endoplasmic reticulum, it is a function as to provide support to colloidal cytoplasmic matrix. It helps in the rapid intracellular transport of the material. Endoplasmic membrane contains a variety of enzymes for various metabolic processes. For example, there is a presence of ATPase and the phosphatase enzyme. The next one we see, it's a Golgi apparatus. The Golgi apparatus, it is discovered by the V. Golgi in 1898 in nerve cell of all and named internal reticular apparatus. The Golgi bodies first observed by the L.S. George. The Golgi bodies, it's a single membrane bound cell organelles. About 60% protein, 40% phospholipid occurs in the Golgi bodies. The Golgi bodies, also named as Golgi body, Dalton's complex, Golgi complex, lipochondria, that is rich in lipid, Becker's body, itosomes, dictyosomes, and the trosporgium. That is, in the dictyosomes means when it is present in the plant, it says as the dictyosomes. This Golgi bodies, it's having the cisternae. There is a presence of the cisternae. The cytoplasm surrounding the Golgi bodies have the fever or the no other cell organelles. It is called as the Golgi ground substance or the zone of exclusion. These Golgi bodies, it is differ in the three form. First one, there is a presence of the cisternae. So this is says as the cisternae. Then there is a presence of the tubules. And there is a presence of vacuoles and the vesicles. The cisternae, these are the unbranched saccules. Like the smooth endoplasmic reticulum, 4 to 8 saccules are arranged on a stalk. Dense opaque material inside the cisternae, it is called as the nodes. This cisternae, it's having the two phases. The first one is a convex phase and the second one is a concave phase. The convex phase, that is says as the cis phase. The convex phase, it says as the cis phase, and the concave surface, it says as the trans phase. This convex surface, that is a cis phase, which is towards the nucleus, and the concave surface means a trans phase, which is towards the membrane. It called as the trans phase or the maturing phase. The second, there is a presence of tubules. These tubules are the branched and irregular tube-like structure associated with the cisternae. Then vacuoles, the larger spherical structure which is present The larger spherical structure which is present, that is called as the vacuole, which is along with the tubules, and the vesicle, it is a spherical structure arise by budding from tubule. Vesicle filled with the secretory material. The next one about the functions of Golgi apparatus. Secretes the material to the intracellular target or outside the cell. Material to be packed as a vesicle from endoplasmic reticulum fused with the cis phase and moved towards the trans phase. This is why Golgi apparatus remain close associated with the endoplasmic reticulum. The protein synthesis by the ribosomes on endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Formation of glycoprotein and the glycolipids is here. The next one we see, that is the lysosomes. These lysosomes, it is a membrane bound vesicle structure formed by the process of packaging in the Golgi apparatus. This lysosome, they are rich in several hydrolytic digestive enzymes, as these are optimally active at the acidic pH. This pH, it is less than 7, therefore are also called as acid hydrolysis and are capable of digesting the macromolecule from various sources like carbohydrate, lipid and the nucleic acid. Generally, these lysosomal enzymes are inactive in nature, but when lysosomes come in contact with another particular organism to form the hybrid structure and it becomes active. After the action of enzyme lysosome, it again reform and reuse. Lysosomes are highly polymorphic cell organisms because during the functioning, lysosomes have the different morphological and the physiological state. They are classified as, the first one is a primary lysosome, secondary lysosome, tertiary lysosome and the autophagic lysosome. The primary lysosome, it is also said as the storage granule. This lysosome stores the enzyme acid hydrolysis in the inactive form. The enzyme synthesis on ribosomes in the cytoplasm. These are newly formed lysosome. The second type of lysosome, it says as a secondary lysosome. The secondary lysosome, that is the digestive vacuoles or the heterophagosomes. These lysosomes formed by the fusion of the primary lysosome and the phagosomes. These are secondary lysosomes. The third type of the lysosome, it says as a tertiary lysosome, or it is also known as the residual body. Lysosomes containing the undigested material are called as the residual body. These may be eliminated by the exocytosis. These are also called as the telolysosomes, means a tertiary lysosome. And the fourth type is the autophagic lysosome or the cytolysosomes or the autophagosomes. Lysosomes containing the cell organelles to be digested are known as the autophagosomes. These lysosomes bring about the intracellular and the extracellular digestion. Afterward, the next one, it's a vacuole. These are the membrane bound space found in the cytoplasm. They contain the water, cell sap, then excretory product and the other material not used for the cell. Vacuole is bounded by the single membrane layer called as a tonoplast. In plant cell, the vacuoles can occupy up to 90% of the volume of the cell. The composition of the cell sap differ in different type of the cell. The cell sap of the central vacuole is a storehouse of the various iron. Thus, it's a hypertonic to the cytosol. Small vacuoles in the seed of certain plants store the organic material like the protein. Vacuoles store the excretory product and even the compound that are harmful, unpalatable to the herbivorous, thereby protected in the plants. Attractive color of the petals are due to the storage of such pigment in the vacuoles. Intake of the food or the foraging particle by the phagocytosis involves the formation of the food vacuoles. In fresh water, unicellular forms like the paramecium excretion and the osmoregulation takes place by the contractile vacuoles which is present in the amoeba. It is vacuoles maintains the turgidity of the cell, means this vacuoles, it is a very important part in the cells. The next one, we see the mitochondria. These mitochondria, these are the clearly visible only when stained. The mitochondria, their number, shape and the size per cell are variable depending on the physiological activities. It is the sausage shape. It may be the cylindrical, or it may be the spherical, oval or the discoidal in shape. The average size of mitochondria is 2 to 6 μm in length and 0.5 μm in diameter. This mitochondria, it's out of which the mitochondria consists of the two membranes. The first one is the outer membrane and the second one is the inner membrane. Out of which the outer membrane, it is a smooth and form a continuous boundary of the cell organism. The inner membrane, this inner membrane, it shows the infolded which is semi-permeable to some metabolites. The inner membrane shows a several finger-like projection. That finger-like projection, it says as a cristae. The cristae is responsible for increasing the physiological activity area, active area or the surface area. The density of cristae determine the intensity of respiration. The cristae having the numerous pig-like structure, and that pig-like structure it called as the oxysomes, or it says as the F1 particle. It is also says as the electron transporting particle. It is also says as a Fernandez particle, or it is Fernandez-Moran particle, or it is also says as the elementary particle. Each particle it is consist of the head region, then the stalk, and this one is the foot region. That is the F1 is here, then there is a presence of the stalk, that is the F6, and the foot is the F0. The head active as an enzyme ATPase synthase, and the foot act as a proton channel. Oxysomes, it is involved in the proton pump and the ATP synthesis. The outer and the inner compartment, it is divided into the lumen into the two aqueous compartments separately. That is says as the outer compartment, and the second one is the inner compartment. The matrix which is present inside to the inner compartment, this matrix contains the single circular DNA molecule and the few RNA molecule. In addition to this, there is a presence of the 70S ribosomes. The mitochondria is divided by means of the fission. The next is about this mitochondria. It's a function. Mitochondria are the site for the aerobic respiration. They produce the energy in the form of ATP molecule. So this mitochondria, it says as the energy powerhouse of a cell. Afterward that, the next one is a plastid. This plastid, these are abundant in all the plant cell and the euglenoids. The plastid, it's a semi-autonomous organelles. They have double membrane envelope. This plastid, it is contains some pigments. The sum of the plastid, it's a large size, easily observable under the microscope. The next one, the plastid, it is based on the pigment. It is classified into the three types. First one is chromoplast, second one is chloroplast, and the third one is leucoplast. That is the chromoplast, chloroplast and the leucoplast. So what's this chloroplast? This chloroplast, these are the plastid which is the green in color. Which is the green in color. It is a greenish in color containing pigment. As this plastid, these pigments are responsible for trapping the light energy essential for the photosynthesis. That is the synthesis of the organic food from the inorganic raw material in the presence of the sunlight. The chloroplast. This chloroplast, it is the play the very important role, that's a photosynthesis. Second one, it's a chromoplast. This chromoplast, it contains a fat-soluble carotenoid pigment like the carotene, xanthophyll. They gives the yellow, orange and or the red color. Means this is mainly present in the cell of the petals, ripe fruit and impart them the various colors. That is, they also synthesize a membrane lipid. The next one is a leucoplast. This leucoplast, it is a colorless plastid, varied in shape and the size and store the nutrient. This leucoplast, it includes the amyloplast, ileoplast and the proteinoplast. Amyloplast, it stores the starch in case of potato. Ileoplast, it stores the oil and the fat, and the aleuroplast, it stores the proteins. Next we see in detail, that's the chloroplast. This chloroplast, it is a double membrane bound organelles mainly found in the mesophyll cell of the leaves. They are the lens-shaped oval, it may be the spherical, discoid or it may be the ribbon-like organelles. Its length is near about the 5 to 10 μm and its width is near about the 2 to 4 μm. The number varies from 1 in case of Chlamydomonas and the green algae and 20 to 40 per cell in the mesophyll. The next, this chloroplast, it is the inner membrane of the chloroplast. The chloroplast, it is shows the consist of the two membrane, outer membrane and the inner membrane. The inner membrane of the chloroplast, it's a less permeable. The space, it's limited by the inner membrane of the chloroplast, it called as the stroma. It contains a number of organized flat-ended membranous sac called as the thylakoids. These thylakoids is present inside the stroma. The membrane of thylakoid, it is encloses a space called as the lumen. The chlorophyll pigment which is present in the thylakoid region. The next is the thylakoid are arranged in a stalk that is called as the grana, and the singular one, it is called as the grana. The grana are interconnected by the intergranal lamellae. That is also said as the stroma lamellae. The stroma contains a small double-stranded circular DNA molecule. There is a presence of ribosomes and the enzymes for the synthesis of the carbohydrate and the protein. The ribosomes of the chloroplast, it is smaller, means it is a 70s than the cytoplasmic ribosomes. As there is a presence of the thylakoid. In this thylakoid means contains the chlorophyll pigment. That is, this thylakoid, it contains the contosomes. The contosomes which contains the photosynthetic pigment. This photosynthetic pigment means the chlorophyll A, B, C, carotene and the xanthophyll. All these pigments are present in the contosomes. So, this chloroplast, it plays a very important role. That is, it helps in the photosynthesis, formation of organic compound, in the consumption of the carbon dioxide and the release of oxygen in the cytoplasm. May also change it into the chromoplast in order to provide the color to the many flowers and the fruit. It also helps in storing the fat and the lipid. Functions is the transduction of energy. The next cell organelles we see as the ribosomes. These ribosomes, it is a protein factories of the cell. It's a protein factories of a cell. They concerned with the protein synthesis. These ribosomes, these are the non-membranous granular structure composed of the ribonucleic acid, means the RNA and the protein. The George Palek first observed the ribosomes in 1953. The ribosomes, it is a of the two subunit. First one is the 16. It's a larger subunit, and the second smaller subunit, it's a 48. Together there is a formation of the ATS, form of the subunit. The next about the cytoskeleton. What's this cytoskeleton? It is a network of the filamentous proteinaceous structure present in the cytoplasm. It provides the mechanical support, motility, and the maintenance of the shape of the cell. The next, there is a presence of the cilia and the flagella. The cilia, these are the hair-like outgrowth of the cell membrane. Cilia, it is a small structure which work like as a hose, causes the movement of a cell or the surrounding fluid. Flagella, it is longer than that of the cilia. It is responsible for the cell movement. Flagella of the prokaryotes and the eukaryote are the structurally different. The cilium and the flagella are covered with the plasma membrane. Their core means this inner side, that is a axonema. It contains many microtubules, and these microtubules, all these are the microtubules. It is running parallel to the long axis. Axonema, these are the 9 pairs. So you can count these 1, 2, 3, 4, 5, 6, 7, 8 and the 9. So these are the 9 pair of the doublets of the radial arranged peripheral microtubules, and one single pair, that is a central microtubules, means it is arranged in the 9+2 array. The central tubules are connected to the bridge and are enclosed by a central sheath. So this one is a central sheath. It is connected to one another. So it is connected to one another. It is connected to one of the tubule to each peripheral tubules by means of the radial spoke. Thus, there is a 9 radial spoke is here. The peripheral doublets are also interconnected by the linkers. Cilium and the flagella emerges from the centriole like the structure called as the basal bodies. Next one is the centrosomes and the centriole. The centrosomes is an organelle containing the two non-membrane bound cylindrical structure called as the centriole. They are surrounded by the pericentriolar material. The centriole lie perpendicular to each other. They are made up of the 9 evenly spaced peripheral fibres of the tubulin. So this is count as a 9. 1, 2, 3, 4, 5, 6, 7, 8 and 9. So there is a presence of the 9 evenly spaced peripheral fibres of the tubulin. Each peripheral fibril, it is a triplet. You can see it's a count as a triplet. The adjacent triplet are also linked. The central part of the centriole is also proteinaceous and are called as the hub, which are connected with the tubules of the peripheral triplet by the radial spoke made up of the protein. The centriole of the basal body of the cilia and the flagella and the spindle fiber that gives rise to the spindle apparatus during the cell division in the animal cell. Next part is a nucleus. The nucleus, it is a first discovered by the Robert Brown. The material of the nucleus, it is stained by the basic dye, was given the name as a chromatin by the Fleming. Normally the cell has only one type of nucleus, only one nucleus is there. Some cells have the more than one nucleus, but in case of the RBC cell, there is an absence of the nucleus. In case of sieve cell, also there is an absence of the nucleus. The interphase nucleus, it contains the organelles like the nuclear envelope, nuclear matrix, chromatin and the nucleolus. So this one is the nuclear membrane, nucleoplasm, that's a nuclear matrix. Then there is a presence of chromatin material and there is a presence of the centrally placed nucleus. The first one is a nuclear envelope. This nuclear envelope, it is a two-layered. It's a two-layer, that is a double membranous component of the cell. The space present between the two membrane of the nucleus, it is called as the perinuclear space. The outer membrane of the nucleus, it is connected with the endoplasmic reticulum at the several places, and the ribosomes are also found on it. Nuclear membrane has a minute pores. There is a presence of minute pore which are the result of the two membrane fusion. The nuclear pore have an octagonal discoid structure as a guard for them, which is made up of the nucleoplasm protein. The pore with the protein structure, it is called as an annulus, or it is known as a bulb. The pore complex which is formed, it is a connection of the nucleoplasm and the cytoplasm. The second, there is a presence of the nucleoplasm, or it is also called as the nucleolymph. Then there is a presence of the chromatin material. This chromatin material, it is a network of the nucleoprotein in the fiber. It contains the DNA and the basic protein, that is says as the histone protein, and there is a presence of the non-histone protein and the RNA. During the cell division, and the chromatin contains to forms the chromosomes. This chromosome, it is stained with the acetocarmine. There is obtaining the two type of region. First one, it is a euchromatin region, and the second one is a heterochromatin region. This euchromatin region, it is a light stained region and diffused part which is transcriptionally and genetically more active. And the second one is a heterochromatin region. This heterochromatin region, it is a dark stained region. It is a thick and the condensed part of the chromatin, having the more histone and the less acidic protein. This part is genetically less active. The next one is there is a presence of nucleolus. This nucleolus, it is a 1 per nucleus. It is the 1 per nucleus. A human cell have the 5 nuclei. The nucleolus, it's a naked. It's a naked and without any membrane, round or slightly irregular part present in the nucleus. It is attached to the chromatin at a specific site called as nuclear organization region. Nucleolus, it is also called as the ribosome factory of the cell, as it has the protein for the ribosome synthesis. So, it is a site for the ribosomal RNA synthesis. The next, there is a presence of the next part, that is says as the chromosome. This chromosome, it is the human cell has a 2 meter long DNA distributed among its 46 chromosome is here, and this 46 number of chromosome, it is present in the 23 pair form. Each chromosome has a primary construction, that is called as the centromere, and every chromosome has a primary construction called as a centromere, on the side of which disc-shaped structure, it is called as the kinetochores. So you can say this one is a kinetochores. On the basis of the position of the centromere, it is of the 4 type. First one, it's a metacentric. Second one, it is a submetacentric. Next one is a acrocentric, and the last one is a telocentric. As in the first one, it's a metacentric. That is a centromere. It is present in the center of the chromosome. So there is a formation of the two equal arm of the chromosome. Second one is a submetacentric. In the submetacentric, the centromere, it is away from the center. So one arm is longer and another arm is shorter. So it says as the submetacentric. Next, there is a acrocentric, means the centromere, it is near to the tip but not exactly the tip. So it is says as a acrocentric. And the last one is a telocentric, means a centromere which is present at the tip region. So it is called as a telocentric. Some chromosomes have the non-staining secondary construction, at the site at a constant location, and that site is called as the satellite. The next, there is a presence of microbodies. These are the membrane-bound minute vesicles that contain various enzymes. It is present both in the plant and the animal cell. So what's the difference between the plant cell and the animal cell? In the plant cell, there is a presence of the cell wall, but in the animal cell, there is absence of cell wall. The plant cell contains a plastid, animal cell shows the absence of plastid. In case of plant cell, there is a presence of the large central vacuole, but in case of animal cell, there is a presence of the small vacuole. Centrioles are absent in case of the plant cell. In case of animal cell, it's a present. The next one is the difference between the prokaryotic cell and the eukaryotic cell. The prokaryotic cell, it is generally smaller, and the eukaryotic cell, it's a larger. Genetic material, it is in form of the nucleoid, but there is a presence of the distinct nucleus. Nuclear membrane, it is absent in case of the prokaryotic cell, and the nuclear membrane, nucleolus, nucleoplasm, all it's present in the eukaryotic cell. The membrane-bound organelles, it is absent in case of prokaryotic cell, but in case of eukaryotic cell, it's present. The circular DNA is here, but there is a presence of the linear DNA in case of eukaryotic cell. The ribosomes, it is a 70S type, and in case of eukaryotic cell, ribosomes, it's a 80S type. Hope so you understood it. Thanks for watching my video. Stay tuned to my channel. Thank